Welcome to Lab 14. So now we're going to be talking about how we identify the human lineage and some of our early members, the earliest members of our human lineage that we have discovered so far. So how do we know? How do we know if we find a specimen? How do we know if it is a part of the human lineage? So several traits have been used as the hallmark indicator that a fossil species is a part of the human lineage, right? So for a very, very long time, the brain size, right? The brain size was considered to be the defining characteristic of humanness, right? However, most researchers today recognize that the emphasis on brain size is problematic for several reasons. So one of the first reasons is that brain tissue does not fossilize. So researchers are forced to work with indirect indicators. So we have a difficulty determining the exact size of the brain. And what we do is we work instead with measurements of a fossil species cranial capacity. So the amount of space in the cranium that exists to allow for a brain. So what is that capacity in there? And this is what we use to estimate brain size. So we can use indentations and ridges on the inside of the cranium to estimate the size of different brain regions, but these estimates, again, are often imprecise and they don't really tell us about the brain's organization, right? It'll give us the size of the brain, the capacity, right? But it won't give us if, say, is the frontal lobe well developed? Is the areas for language well developed, and so on and so forth. It doesn't tell us anything like that. Secondly, having a large brain is not actually unique to humans. Brain size is generally proportional to body size, although we have a large brain relative to our body size, but this is also not unique. And then third, an increase in brain size to modern human range is a relatively recent evolutionary step in our history. Therefore, if we were to use brain size as our sole indicator of a species belonging to the human lineage, we would be missing a large part of our own evolutionary history. A lot of the species that we consider now as part of our human lineage would actually be excluded from our human lineage because their brain size is not that large. So, another popular trait that we have used in the past to distinguish our lineage is tool use. Like the brain size, this is also problematic. This approach has traditionally emphasized stone tools because they preserve well and they're visible in the fossil record. But what this does is it downplays the potential significance of tools that are made from less durable materials, things like plants. And they then may inadvertently exclude from our lineage numerous species that use plant-based tools. So, research has demonstrated widespread tool use in non-human apes and some monkeys. So if we were using tools as an indicator of being a part of the human lineage, then this is also a complication. So what it does, right, our research shows, it suggests that tool use is not limited to humans and our closely related ancestors. Rather, tool use is a more common behavior of primates in general. So given the problems with using brain size and tool use, a different characteristic needs to be used to identify members of our lineage. So the best characteristic for identifying members of our lineage is bipedalism. And bipedalism is a form of two-footed locomotion, where movement occurs primarily through the use of legs. It's an unusual form of locomotion among primates, and it appears very early on in our evolutionary history. So it's not as simple as just bipedalism. There are types of bipedalism. So it defines our lineage. It is not restricted to our species, though. So many primates practice some sort of bipedalism. What distinguishes us is that we practice obligate bipedalism. Aside from obligate bipedalism, there are two other kinds of bipedalism. So we have occasional and habitual bipedalism. Occasional bipedalism is bipedal locomotion that is practiced sometimes or occasionally. Occasional bipeds will rely on a different form of locomotion as their primary means of movement, and they often fall back on bipedalism under certain circumstances. So, for example, Non-human apes will move bipedally on the ground when they are using their hands to carry things. So they are occasional bipeds. So because the primate rarely practices bipedalism, it has few, if any, anatomical adaptations for its primary form of non-bipedal locomotion. Then we have habitual bipedalism. And habitual bipedalism is bipedal locomotion that is practiced regularly or habitually. Habitual bipeds may also rely on a different form of locomotion, but they use bipedalism and their other locomotion strategy more equally. So, for example, many of the early members of our own lineage practiced habitual bipedalism. The Australopithecines that produced the Laetoli footprints were habitual bipeds. They were still somewhat arboreal, and they spent a lot of their time climbing in trees. On the ground, though, they often practiced bipedalism. So these species used bipedalism more often than an occasional biped, but they also maintained and used other forms of locomotion. So habitual bipeds we see have a mix of locomotor adaptations. They are bipedal enough that they have some selectively favored bipedal adaptations. However, they also use an alternate form of locomotion, such as arboreal climbing, enough that they have some selectively favored adaptations for that locomotion as well. And then we have obligate bipedalism. This is bipedal locomotion that is practiced all the time. Our obligate bipeds have no other realistic locomotor option. Humans today are obligate bipeds, as were all of our ancestors since Homo erectus about 1.8 million years ago, and perhaps even Homo habilis about 2.5 million years ago. We can cover short distances when we're crawling on all fours, and we can climb trees, and we can practice arm-over-arm swinging. However, we cannot do these things comfortably for any length of time without special training and practice. Once we reach a certain age and body size, we're anatomically obliged to walk on two legs, right? So just think about yourself, right? If you have to crawl anywhere for an extended period of time, if you have to practice arm over arm swinging, climbing trees, how long can you comfortably do that? I know personally, specifically with arm over arm swinging, I have very, very little upper arm strength, so that won't last me very long. Right, so we can knock out climbing trees also because, again, very little arm strength. Crawling, probably for a little bit, but then again, you can only do that for so long and still be comfortable. So we have to eventually then get up and walk on our two legs, unless there's other reasons why. So anatomically, though, we are obliged to walk on our two legs. So, however, I'm trying to say is that we still maintain those few adaptations for other forms of locomotion, right? The fact that we can, cross the monkey bars, right? That's the fact that we have maintained suspensory traits, right? Most of our locomotor adaptations, though, are for bipedalism. Our bodies are more suited for bipedal locomotion than any other alternative. And when we analyze the fossil record, we examine the physical traits of a fossil species. We then use the data to then make a hypothesis and interpret the behavior of the species as a whole. So when we are trying to identify if a fossil species was a member of our lineage, we look for anatomical traits for bipedal locomotion. Bipedal anatomical traits indicate that a species was at least habitually bipedal, and it practiced bipedalism frequently enough that natural selection favored those traits and facilitated this locomotion. These habitual bipeds are more bipedal than members of non-human primate lineages and therefore belong in our human lineage. So what are these bipedal adaptations? And you're gonna have to know them. You're gonna have to know all of the bipedal adaptations. Right, so there are numerous adaptations for bipedalism that can be seen in our skeleton as well as in the fossilized skeletons of our ancestors. So I break it up into sections. Is how it's going to be broken up on your quiz into sections. You're going to have a written portion where I'm going to ask you to name a bipedal adaptation in each of these sections. So fair warning. So two important bipedal adaptations found in the cranium. The first is the position of the foramen magnum, and the second is the mastoid process. So remember, the foramen magnum is the large hole at the base of the cranium that allows the brain to connect to the spinal cord. So it makes sense that the position of the foramen magnum will be positioned differently in a biped versus any other locomotor adaptation. We can see here, right, visually, in humans, the foramen magnum is on the bottom of the cranium. It is here, and then your spinal cord goes downwards, right, perpendicular to the ground. And then the position of the foramen magnum in a non-human ape, monkeys, and other quadrupedal animals is towards the back of the cranium, where then it connects to the spine, and the spine runs parallel to the ground. So in a quadruped, the foramen magnum is positioned more posteriorly, or towards the back of the cranium. This allows then the vertebral column to remain parallel to the ground while extending outward from the brain case. In a biped, the body is oriented more upright, and the vertebral column is perpendicular to the ground. Therefore, the foramen magnum of a biped is positioned more anteriorly towards the front of the cranium and inferiorly on the underside of the cranium. So what this does is it helps center the head over the upright vertebral column. Then we have also the mastoid process. And so the mastoid process, remember, is a triangular bony projection located posterior to the ear, and it allows for the attachment of neck muscles. Remember, I told you when we were talking about the mastoid process during the bones lab that it was going to come back later in the semester for bipedal adaptations. So bipeds have a larger mastoid process than a quadruped, and it reflects the differences in their neck muscles. In bipeds, the head is centered over the body, and there is a lot of potential for anterior, posterior, and lateral head movements. Because of this, we have numerous relatively large neck muscles that help hold it in place. The mastoid processes are areas of muscle attachment for several of these neck muscles, and they are large in bipeds to accommodate these muscles. So those are the two adaptations we find in the cranium. The next major bipedal adaptation is found in the vertebral column. So bipeds have an S-shaped vertebral column with curves in the thoracic and lumbar areas. In the thoracic region, the vertebral column curves back, creating a convex dorsal or a posterior surface. And then in the lumbar region, the vertebral column curves forward, creating a convex ventral or anterior surface. When you look at it from the side, it resembles an S, so we say the S-shaped vertebral column. In non-bipeds, the vertebral column is straighter with only one curve that gradually forms a long, convex dorsal surface. Then, if we're looking at the vertebral column, also in a biped, we have the presence of the lower lumbar curve in bipeds, which helps shift the upper body weight forward and center it over the pelvis and lower body. In addition, bipeds have large lumbar vertebrae. These large lumbar vertebrae help support the weight of all the body parts above them. So they have large vertebral bodies to accommodate this function, right? So adaptations in the vertebral column are the S-shaped curve and the large lumbar vertebrae. I group in the pelvis into the midsection. So when I break it up on the quiz, I'll ask you cranial adaptation, midsection adaptation, and then a lower appendage adaptations. So the pelvis of a biped is also specially adapted for bipedalism and has an unusual bowl shape. That bowl shape is largely the result of the differences in the two ilia. In a biped, the ilia are short and broad, and they are positioned more laterally in the pelvis. So really quickly, I'm going to show you this image here, but I want to explain what is going on here and why that shape difference exists. In non-human apes, if you're looking up here, right, this is from a non-human ape, the gluteus medius and the gluteus minimus muscle pull the leg backwards, which extends the leg at the hip. In humans, the medius and the minimus gluteal muscles abduct the leg. That is, that they pull the thigh away from the midline of the body when the leg is extended. As the thigh is abducted, it also rotates inwards and provides a more stable support for the hip during walking. So we have a different pelvic structure to reflect that. Okay, and just really look at this here. And I want you to just even think about it. If you're thinking about, like, a cat or a dog, their legs muscles, their legs just in general, go forward and backwards only, right? That abduction is kind of, it is a difficult move for them to do because they don't have the muscles built specifically for that movement. Whereas we can abduct our leg outwards quite easily because our muscles allow us to do that. And we have the bones, the bone structure to allow for that. Okay. So this shape is related to the differences in the leg muscles of bipeds. So while we're familiar with the gluteus maximus, we are less familiar with the two related muscles, which are the gluteus minimus and the gluteus medius. These attach to the ilium on one end of the femur and on the other. In quadrupeds, these muscles straighten the thigh and they act as powerful extensors for quadrupedal locomotion. In bipeds, these muscles perform a very different function. So you think about it, when we walk, we only have one foot on the ground at a time, right? We have to balance on one foot as we make a stride. So the foot and the leg need to help to remain steady and balanced during walking. In bipeds, the gluteus maximus and minimus serve to stabilize the pelvis when all the weight of the body is on the balancing leg. Because these muscles serve a different function in bipeds than quadrupeds, they have a different shape and position. Remember, when we have muscles in different shapes, different positions, then the bones are going to reflect that. So the biped pelvis is shaped differently to accommodate those unusual gluteal muscles. Overall, the bowl-shaped pelvis and corresponding gluteal muscles helps us to maintain a stable and efficient bipedal gait. In addition to muscle attachment function, the bowl-shaped pelvis also helps support our intestines and other organs when our bodies are positioned upright. So, going over bipedal adaptations. Cranial adaptations include the position of the foramen magnum underneath at the bottom of the cranium, you can just say that, or a larger mastoid process. In the midsection, we have the S-shaped vertebral column, larger lumbar vertebrae, and a bowl-shaped pelvis, right? So those are the ones we've covered so far. Now we're getting into our lower appendages. So compared to our other living relatives, we have much longer legs. This is largely the result of our elongated femurs, tibias, and fibulas. In addition to being longer, the biped femur also has an unusual angled position. The femur, as we can see here, angles inwards as opposed to that of a non-human ape. So in most primates, that femur will extend relatively straight from the pelvis to the knee. What it does is it keeps the knee towards the lateral side of the body. Bipeds, however, have an angled femur, and that angled femur extends down and medially from the femur to the knee. This centers the knee and the lower legs underneath the body and near the midline, which then improves our balance and walking efficiency. As we move forward, our body will follow that central path rather than wobbling from side to side. In addition, when we briefly balance on one foot, each time we take a step, the lower leg is closer to our midline and more stable. So those are the legs. We also have adaptations in our feet. So the feet support the entire weight of the body, and they have several key adaptations because of this. Firstly, bipeds have short toes, right? We don't use our toes for grasping. Instead, we use our toes for support while standing and to help push off when we step forward. This helps with bearing weight and transmitting the force as we take steps, right? Especially that big toe being larger than our other toes, which is the opposite of what we see in other primates. Secondly, bipeds have a non-divergent hallux, and that's your big toe. That is in line parallel with your other toes. So that non-divergent hallux is very, very important for balance and pushing off. Thirdly, the biped foot has a longitudinal arch that extends down the length of the foot from the base of the toes to the heel. So that longitudinal arch helps with the shock absorption as we walk, and it helps to direct our body weight and energy to create smooth push off when we're stepping forward. So in the feet, we have short toes, a non-divergent hallux, and a longitudinal arch. Those are the 3 adaptations in the feet. So let's really quickly go over everything again, and then we'll discuss why bipedalism evolved. So in the cranium, we have the foramen magnum positioned more anteriorly and at the base of the cranium, inferiorly. We also have larger mastoid processes. In the midsection, we have an S-shaped curve in our vertebral column and larger lumbar vertebrae. We also have a bowl-shaped pelvis. In our lower appendages, we have femurs that angle inward, medially. And then we also have short toes, non-divergent hallux, and a longitudinal arch in the foot. So remember all of these. They'll come up in your labs. They'll also come up in your quiz. So here is a human foot in comparison to the chimpanzee foot. And here's again a medial view of the human foot versus the chimpanzee foot. So why did bipedalism evolve? There are numerous explanations that have been put forward and have gained a varying amount of support in the scholarly community. Most famous of these explanations is the savanna hypothesis, which is supported by Raymond Dart. Raymond Dart is an early paleoanthropologist who first identified Australopithecus africanus. The savanna hypothesis argues that bipedalism was selectively favored as open grassland environments expanded throughout Africa. Around 8.8 to 5 million years ago in Africa, many of the forested areas shrank and were replaced by savanna environments, which are open grasslands with interspersed pockets of trees. The shift to savanna environments was further facilitated by geological activity in eastern Africa. So at the time, we had large tectonic plates that were splitting apart or rifting. This caused the landscape changes that contributed to more dramatic differences between the wet and the dry seasons. These seasonal changes, combined with overall drying, encouraged the expansion of grassland habitats in the region. So according to the savanna hypothesis, bipedalism evolved as a favorable adaptation that facilitated life in these grasslands, where earlier forms of arboreal locomotion would be less helpful. The savanna hypothesis was supported by researchers for many, many years, but since the 1990s, anthropologists have discovered numerous fossils of the earliest members of our human lineage, which challenge the validity of this explanation. Many of the earliest species may not have lived in savanna grasslands at all. So, alternative hypotheses for the evolution of bipedalism, such as the provisioning hypothesis, must be considered as well. Like the savanna hypothesis, we have the patchy forest hypothesis. It ties the evolution of bipedalism to environmental changes. This theory was put forward by Peter Rodman and Henry McHenry. Rodman and McHenry argue that bipedalism would have been more energy efficient than quadrupedalism as the environment in Africa transitioned to a patchy forest over several million years. So from the end of the Miocene to the Pliocene is what we're talking about. And so today, bipedal humans are more energy efficient in these types of environment than our quadrupedal chimps. But it's unclear if the first bipeds would have had the same efficiency benefits that we do. As we see later in Lab 15, the transition to obligate bipedalism is not as clear-cut. So the earliest bipeds in our lineage would probably not have benefited from an increased efficiency when they were walking upright. So another explanation based on environmental changes is a thermoregulation hypothesis, and it was proposed by Peter Wheeler. And so this hypothesis argues that bipedalism was favored because it helped our early ancestors manage their body temperature and stay cool during the heat of the day. So This says that an upright posture will lift the body into a slightly cooler zone of air above 3 feet off the ground. This then exposes the body to more wind and air circulation, which helps with cooling down the body. Critics of this theory have noted that the earliest hominins lived in environments that still afforded plenty of tree coverage, in which they could find shade during the hottest times of the day. So that upright posture was not really necessary to regulate the body temperature. Which then brings us to the provisioning hypothesis. And so the provisioning hypothesis argues that bipedalism evolved in early humans that were monogamous, specifically to allow male human fathers to provision food and resources to the human female partners. This would ensure them the survival of their offspring and secure mating access to mothers. The provisioning of females required males to have free hands for carrying food, so bipedalism is argued to have evolved to facilitate this. However, the validity of this hypothesis has also been challenged, particularly as it depends on the social structure, specifically a monogamous social structure of early humans, which is not clearly known and we can't actually observe it. So, in the final example, the final theory, we have the postural feeding hypothesis that was just presented by Kevin Hunt and is also based on the feeding benefits of bipedalism, although it does not assume provisioning or particular social relationships. So the postural feeding hypothesis argues that an upright posture helps apes to more efficiently reach hanging fruit in small trees typical in African woodland environments. The earliest bipeds in our lineage became upright not because of walking then, but because of standing to collect these fruits. Once our ancestors had adapted a few traits that helped with balancing in an upright posture, additional traits that helped with walking upright became favored as well. What began as an adaptation to slowly shuffle from one tree to the next eventually became the full bipedalism that we know today. However, it's unclear how much of the diet would have come from trees and these fruits. So therefore then how important the ability to collect them would have been among our ancestors, that also is unclear. Right. So again, these are all theories, right? But we're not entirely sure. We don't have the answer specifically for which is the best theory. So now let's talk about our earliest ancestors. We're just going to cover Ardipithecus, really. Or Australopithecus. Australopithecus? Yeah, Australopithecus. So, Australopithecus. We are covering Ardipithecus. Australopithecus. For almost 100 years, we believe that the first members of our lineage were a group of fossil species in the genus Australopithecus, right? And that's why I got confused here, because I thought we're starting there. We're actually going back. So since the 1990s, 4 new fossils have been identified that possess the bipedal adaptations unique to members of our lineage. They lived between 7 and 4.4 million years ago, which predates our Australopithecus species. So they are called our pre-Australopithecus, our pre-Australopithecines. They have a mix of adaptations for bipedalism and arboreal climbing. So this shows that the evolution of bipedalism was probably gradual and occurred in a mosaic fashion, with different bipedal adaptations evolving separately over time instead of simultaneously. So first we have Sahelanthropus chilensis. This is the first of the pre-Australopithecine species. It lived around 7 to 6 million years ago in the modern Central African country of Chad. Sahelanthropus chilensis has a pronounced brow ridge and a small cranial capacity of about 350 cubic centimeters, similar to a living non-human ape. It is argued that its foramen magnum is positioned more anteriorly, which indicates more human-like bipedal locomotion. Sahelanthropus chilensis also has a relatively smaller, less prognathic face than non-human apes. So this mixture then of facial and bipedal traits, as well as the early date for Sahelanthropus, suggests that it may have been the earliest member of humans and other apes such as chimpanzees. Then we have Aurantugenensis, around 6 million years ago. It was discovered in the year 2000 in the Tugen Hills of Kenya. It is not confirmed how Auroran is related to modern humans, and as of 2007, 20 fossils of the species have been found. We have the posterior part of a mandible in 2 pieces, a symphysis and several isolated teeth, 3 fragments of femora, a partial humerus, a proximal phalanx, and a distal thumb phalanx. Auroran also had small teeth relative to its body size. So while its teeth are more similar to those of non-human apes than humans, it appears to have had adaptations for bipedalism. Although the available fossil fragments cannot be used to identify if their femurs were angled, the available proximal ends of a few Auroran femurs have bipedal traits. The femurs have long necks as well as a groove for the obturator externus muscle, which helps stabilize and rotate the leg. Both of these features are seen in modern bipedal humans, but we don't see it in modern chimpanzees. And so you can see here in the image, we have modern human, we have Auroran, and then we have a chimpanzee. And so we have that long neck in both of these for the location of that obturator externus muscle to go through. And so we can hear a chimpanzee has a short neck, so no obturator externus groove. Then we have the last 2. So the last 2 pre-Australopithecines have both been classified as members of the Ardipithecus genus. So we have Ardipithecus cadaba as the older of the 2 species. And the other one should be Ardipithecus ramidus. Yeah. But Ardipithecus cadaba is the older of the 2 species, and it was around 5.5 million years ago. The teeth appear to have some modern human-like features and some chimpanzee-like features, and its early date makes it a contender as a possible ancestor of humans and chimpanzees. So you can see here on the right, we have Ardipithecus cadaba teeth. Ardipithecus cadaba had a honing canine, which is similar to what we see in chimpanzees today, which is what we see here on the left. The honing canine is naturally sharpened by rubbing against the lower premolar when the mouth is opened and closed. So that's what this honing canine is. Okay, so that's Ardipithecus cadaba. Then we have Ardipithecus ramidus. It's more recent, so 4.4 million years ago, and the most famous specimen is one we have nicknamed Ardi. Ardipithecus ramidus had a small cranial capacity and a short stature of around 4 feet tall. Ardipithecus ramidus also had very long arms and fingers, but other upper limb features indicate it probably practiced more of an arboreal climbing than a suspensory locomotion. Bipedal adaptations we find here are a shorter and wider pelvis. The feet have a mixture of locomotor adaptations with long toes and the hallux being divergent, suggesting that the feet were still frequently used for arboreal grasping. However, the feet also show clear signs that they were used for pushing off in bipedal walking. So as a group, the pre-Australopithecine species share 2 important things in common. First, the pre-Australopithecines are habitual bipeds. They have a mix of adaptations for bipedalism and arboreal locomotion. And the second, based on the analysis of animal and sometimes plant remains from pre-Australopithecine sites, we know that they lived in forested environments. So when we take this together, these features directly challenge that long-standing savanna hypothesis. The pre-Australopithecines were bipedal in a forest, so the evolution of bipedalism was not the result of living in an open savanna grassland. Rather, bipedalism must have evolved for a different reason. Right, so like I said, the theories, they're still theories. They haven't been proved yet, and I don't know whether they will be ever proved true or not. I mean, I think we need more specimens to analyze in order to really say we have proven something. Right. This is a video that kind of summarizes this evolution of bipedalism in our early ancestors. Please take a look at it. And we'll be having more videos embedded in the last 2 labs as well. But please take a look at it. It does a good job of explaining, especially the earlier Australopithecines. But that's it for this lab. Next week, we are moving completely into the human lineage, going from our Australopithecines and then into the genus Homo, and then all the way to modern humans today. But that's it for this lab in this week.